All right, hello. Sorry I can't be there today. The notes that we're going to be going over today are titled Chromosomes. Please write this down on the top of your notebook. Chromosomes. What we're going to be looking at is the information that's found in Chapter 8, Section 1 of your textbook. So if you have anything that's a little bit unclear as we go through these notes, please feel free to go and take a look in your textbook at Chapter 8, Section number 1 later on as you get home. First thing that we're going to be looking at here, definition. So draw that line, write down your right side of your paper, just like we've been doing the last several sheets of notes that we've done, not all the way into the middle, but about a third of the way over. On the left side of that line, on your right page, write that word chromosomes right there. And the definition of what a chromosome is, it is a long, organized strand of DNA. So long, organized strands of DNA. We have lots and lots of these chromosomes in our cells. We're going to be looking at exactly what makes up these particular chromosomes and kind of how it functions in our cells. Chromosomes. When we take a look at these long strands of DNA, what we've noticed, what scientists have figured out, is that particular sections of these long strands of DNA are made up of what we have termed genes. So underneath chromosomes on that left side, write that word genes. And our definition here for what a gene is, it's a part or section of a chromosome that codes for a trait. That is, it codes for a particular protein. So genes are those parts, they're sections, and that goes on the right side of chromosomes that code for a particular trait. Now when we take a look at these chromosomes, what we find typically when we look at the chromosomes is a picture that looks like this. And this actually is a photograph. And it's a photograph of what we call a pair of these two chromatids. So here's one chromosome over here. And here is another chromosome right here. It's an identical copy. To kind of differentiate between just what a chromosome is by itself and what we can actually see, we see that we have these chromatids. So on that left side, write down a chromatid, and on the right, it's an identical copy of chromosome. Identical copy of a chromosome. And the reason we talk about these chromatids is because this is the time when we can actually see these particular structures in our cell. We have a very difficult time still, even with all of our scientific improvements in microscopes, in actually looking at our DNA when it's just a single chromosome. We can only see it when it's what we call this identical copy. So after these, this chromosome has copied itself, we see them in these forms called the chromatids. Now these chromatids are held together in the middle at a location called the centromere. This is the place that holds these chromatids together. That centromere is located right here. It's kind of this area where it's pinched in a little bit. It's not necessarily a thing, it's more of a place. So on the left side, write centromere. And on the right, it's the middle that holds these chromatids together. So we have one single chromosome, an identical copy, makes two chromatids held together here at the centromere. Centromere. Now when we look at our cells, when we look at the particular chromosomes that are in our cells, what we find is we have a couple different types of cells. We have one type of cells that are called the diploid cells, and one type that is called the haploid cells. So let's look at the diploid cells first. So on that left side of your line, write down that word diploid. And then our definition for diploid, di meaning two. So these are cells with two of every chromosome. You'll also see diploid cells called the 2N, where N represents the number of chromosomes. Now, different organisms are going to have different numbers of chromosomes. But organisms are all going to be these diploid particular cells. They have two of every single chromosome in them. Cells that, for us, that are diploid, a couple examples. So on the right-hand side of your line, write down example. Then write down normal cells, things like our muscle cells, skin cells, the cells that make up your lungs or your stomach. These are our diploid cells, our normal cells. There's another term for these diploid cells. It's this term somatic cells. Somatic cells. That's another term for the normal cells of the body. In a human, that would be all of us, our diploid cells have 46 total chromosomes. So, if diploid cells are cells with 2 of every chromosome, that means we have 23 pairs of chromosomes making up our diploid cells to make that 46 total chromosomes. 46 total chromosomes in our normal body cells, of which we have somewhere right around that 2 trillion mark. Our haploid cells, they write down haploid on the left side of that line, these are cells with 1 of every chromosome. So this is different. These are called the 1N. One way to remember this term haploid cells is that they are half of a diploid. So that HA in haploid and the HA in half. They have half of the number of chromosomes that are in a diploid cell. The haploid cells for humans are only our egg or sperm cells, depending, of course, on whether or not you are a male or a female. Just the egg or the sperm cells make up our haploid cells. No other cells are those haploid cells. Another name for these egg or sperm cells, for these haploid cells, are the gametes. So, AKA, also known as the gametes. So you have a few different terms on this particular page to worry about. Diploid cells are our normal cells, also called the somatic cells. And there's 46 total chromosomes in a human diploid cell. Haploid cells have half of that. They're our egg or sperm cells. We call those the gametes. So very important information here with these particular terms to make sure that you have dialed in very well. So our gametes, because they are half of a diploid cell, we take that 46, and in our egg or sperm cells, we have 23 total chromosomes. So that's half of our normal cells. And that plays a very big role in what we're going to be talking about here in this unit on reproduction. So, think about how you were made. No. Don't think about how you were made. Put that thought out of your head. Seriously, don't think about that. Just be thinking actually about an egg cell and a sperm cell. And this egg cell and the sperm cell have to meet in some way. So when this egg cell, which is a gamete, has 23 human chromosomes in it, this sperm cell, another gamete, has 23 human chromosomes in it. When they come together and the 23 chromosomes from the egg and the 23 chromosomes of the sperm meet, we now have, wait for it, 23 plus 20, 46 total chromosomes in that very first cell that was you. So each of your parents, so on the left-hand side, write down how I was made. And then on the right-hand side, each of my parents contributed 1 of each of their chromosomes. So they contributed 23. That's from these 2 haploid cells. So each parent contributes 1 of each of their chromosomes. And when this big process called fertilization happens, the egg and the sperm, again 2 haploid cells, combine together to give you 46 total chromosomes. So here at this fertilization process, egg and sperm, 2 haploid cells, combine to give you 46 total chromosomes. Now that's just for humans. Other organisms are different. Dogs don't have 46 total chromosomes. They have other numbers. We'll get into those a little bit later on. But in any case, when we're talking about this sexual reproduction, an egg and a sperm coming together, it's a haploid cell and a haploid cell. It doesn't matter exactly what organism it is, as long as it's 2 haploid cells combining to give us that total of these 46 chromosomes. Once we have an egg and a sperm combining together to make those 46 total chromosomes, we have a new cell type. And this cell type is called a zygote. And that is the beginning of the new offspring. You were a zygote, which was one cell with 46 total chromosomes inside of that one cell. Zygote, the new offspring. Like I mentioned before, 46 total chromosomes. Another way of putting that, a little bit easier, is that now this offspring, this zygote, is one diploid cell. Now, we can, as scientists, actually look at our chromosomes, and we call that a karyotype. So on the left side of the line, write that word karyotype, and then on the right, here is the definition. It's a picture of all chromosome pairs in a diploid cell. Remember, you got 23 from mom and 23 from dad. Your dad is giving you chromosomes that have the genes for your eye color, and your mom is giving you chromosomes that have genes for your eye color. That's all on the same chromosome. So we have these pairs of chromosomes. In order for us to look at these, scientists need to actually reach into a cell, pull out these particular chromosomes, and then organize them. So when we organize these particular chromosomes, we're actually looking at what we call the homologous chromosomes. Homologous. Now this particular prefix homo is one we're going to be using quite often, and it's probably one you're familiar with. Most of us here in America are familiar with the term homosexual. Homo means same, and sexual, sex. And someone who is homosexual likes the same sex. Someone who is heterosexual, we talked about hetero in our last unit, heterotrophs, different or other. So males and females. So here, homologous chromosomes, these are the same chromosomes. So homologous chromosomes underneath karyotype on the left side, and our definition here might be a little difficult for you to see. These are the pairs of chromosomes, one from each parent. So when we look over here, and this is a karyotype, we have these numbers 1, 2, 3, 4, 5 across the top, and then 6 through 12, 13 through 18, and 19 through 23. So here's a karyotype right here. What we'll find is that there are two pairs up here, and two pairs here, and two pairs here, and two pairs here. One from mom and one from dad. Now, we can only actually see these chromosomes after they've made the chromatids, those identical copies. So we have one chromatid and another chromatid. This one came from your mom. And here's a chromatid and another chromatid. This one came from your dad. And from mom and from dad, mom, dad, mom, dad, mom, dad, mom, dad, mom, dad. So each pair has one chromosome from mom and one chromosome from dad. Now you might be thinking, well, why are we even talking about these karyotypes? Why do we need to look at our DNA? Well, scientists, doctors, and parents use these particular pictures of our DNA to determine some information. So, on the left-hand side, use of karyotype. Use of karyotype. And then on the right, by scientists, doctors, parents, to determine gender. We can actually figure out what the gender is of our unborn child before that ultrasound can tell us. We also use it to determine if there's any abnormalities, if there's any differences in these chromosome numbers that's going to have a significant impact on the individual that has those particular chromosomes. Some of you might be familiar with something called Down syndrome. Down syndrome individuals have one extra chromosome. That's it. Happens to be one of the chromosomes down here. We'll talk about that here in our next class period a little bit more in detail. That's it, just one extra chromosome, and we have significant changes in how a particular individual can react to the outside world. We have two particular kinds of chromosomes that we look at in these karyotypes. First one here are the autosomes. These are our normal chromosomes. And in a human's case, when we look at a karyotype, the autosomes, and write that word autosome on the left side of the line, and on the right side it's the normal chromosomes. The autosomes are chromosomes numbers 1 through 22. Those are all of our normal chromosomes. And then we have some special ones that are down here. And these special ones down here are our sex chromosomes. Left side of the line, sex chromosomes, and on the right, these are the chromosomes that determine our gender. Whether we are a male or whether we are a female is determined by our cells. Every single cell that we have in our body has these sex chromosomes in them. Whether it's a cell that makes up the skin on the back of your neck, or it's a cell that makes up the lining on the inside of your stomach. Whatever it might be, that cell is going to have all of these chromosomes, including these particular sex chromosomes. And the sex chromosomes that determine our gender are as follows. This goes on the right side. If you are a female, 2 X chromosomes, so XX equals female on the right side. If you are a male, 1 X chromosome and 1 Y chromosome. That is a male. So the only thing that makes guys so very different is that we have these Y chromosomes and females do not. That's enough to have huge, enormous differences in the particular proteins that our cells are making. And then on the right-hand side, the last part here, all humans, normal humans, have 22 pairs of autosomes and 1 pair of sex chromosomes for 23 total pairs of chromosomes. So 23 pairs. And since there's 2 in a pair, that means we have 46 total chromosomes in each one of our cells. So on the right, humans have 22 pairs of autosomes, 1 pair of sex chromosomes, which equals 23 pairs, or 46 total. Now, you're going to be receiving this sheet in just a moment. You are going to be just like a scientist. Scientists reach into a cell, open up the nucleus, pull out all of this DNA, these chromosomes, and they're in this big mass just like this. And then what they have to do is they have to lay those chromosomes down on this plate, and they have to actually pull them apart and organize them. You're going to be doing the same thing. You're going to be taking this mass of chromosomes, and you're going to be pulling them apart. You're going to be using scissors, which are located at each of the lab stations in the little plastic containers. Each of you will be cutting each one of these particular chromosomes out. You'll be doing this solo at a lab station. Once you've cut all of these out, you're going to need to take a piece of binder paper. You're going to need to lay that piece of binder paper on the lab station, and you're going to need to put all of these 46 chromosomes in order. What's the order? Here's the order. Largest to smallest. That's all you have to go by, is largest to smallest. One more time. Largest to smallest. That's all the scientists have to go by when they're manipulating the real thing, is they take the biggest ones and they put the biggest ones first, and then they find the next biggest ones, the next biggest ones, the next biggest ones, the next biggest ones, until they get to the smallest ones. That is the karyotype. That is going to be your task now. So you'll be getting this, cutting them out, organizing them from biggest to smallest on a piece of binder paper, and using the glue sticks, I'd like you to glue them down. But make sure you have them from biggest to smallest. Don't lose any. We're going to be utilizing this karyotype in a couple different activities throughout this unit and next semester. So make sure that you take a little bit of time to get this information in perfectly. If you have any questions, please feel free to raise your hand. It shouldn't be that difficult. I didn't say anything about the letters. Just organize them from biggest to smallest. Fantastic. Don't lose any. Whatever you don't finish, find a way to take them home and get it done by next class period. Great.